Start recording. There we go. Share my screen. I may as well do that too. State that there was no lab scheduled in Zoom today, so I had to create one. And I imagine some of you students are not going to be able to join us for the lab, but I will make it available on YouTube. What I'm going to be doing is just finishing the review for the final. Any questions by those of you who are here? How many students do I have? My computer is a little slow because I'm still working on the Zoom session, 93% done for the lecture. I'm going to stop the share for a minute. Hello? Is anyone there? Yeah, there's 2 students there. All right, let's
Yep, we're here.
share again. All right, let's begin. I have emailed Roberta about the Zoom sessions, and hopefully I'll get an answer to that. All right, let's get going. Chapter 8 was a very involved chapter, but you've already been tested on it. So there will only be a few questions from chapter 8. Let me mention you should study the lab. I'm not really going to review the lab further, but you should know at least the main point of each lab and be able to discuss that on a quiz question. And you should know the different terms for the labs. What else? You should know the general lab procedures for how we streak out a plate for colony isolation, how we use aseptic technique to prevent the plate from getting contaminated or from preventing the tube to get contaminated. You should know a little bit about the founding fathers of microbiology, especially Louis Pasteur, who started the aseptic technique, aseptic culture, keeping the culture pure, and then starting a pure culture. He did that with a liquid culture, and it's very difficult to get a pure culture using liquid culture. He'd use limiting dilution, which I think I've talked about previously. It became much easier when Dr. Koch invented growing bacteria on a solid media and then just streaking on over a plate, getting isolated colonies. Now, to get a pure culture, you need to pass the cells through 2 colony isolation events where you streak the plate for isolated colonies, not once, but 2 times. Any question about any of that? And then you should know about where you put your material in the lab. Obviously, you didn't go to the lab, but you bring in everything that should go in the cubby holes. And by the door to the cubby hole, we consider that area outside of the lab. Anything else that comes into the lab proper should stay in the lab, well, forever. So any pencil you bring into the lab should stay there. And you should not take it home until it has been disinfected, either by being autoclaved or put in a bucket of disinfectant. And you should also know about where you should put waste material, like your Petri dishes, when you are done using them, where they should go, where you dispose of material, where paper towels go if they have bacteria contamination on them, where paper towels go if they do not have bacteria contamination on them, but they are waste. Any question about any of that? Why am I here? I'm going to close that one down. We've already talked about chapter 14, right?
Yes.
All right. All right, chapter 8, microbial genetics. Be able to define the terms genome, chromosome, gene, genotype, phenotype. Be able to describe vertical and horizontal gene transfer. So vertical gene transfer is from the gene transferring from the parent cell to the offspring cell, or the parent to the offspring. Horizontal gene transfer is where a gene moves from one cell into another cell of the same generation. And there are 3 ways that horizontal gene transfer can happen in bacteria. Let's skip down to talking about that. Here it is. Horizontal gene transfer can happen by transformation, conjugation, and transduction. In humans, we don't really have conjugation. I mean, we have reproduction, but it's not conjugation. We do have transformation, although human cells do not readily take up DNA and then become transformed. But bacteria cells do. And then transduction probably happens in our nose all the time, but it's unimportant to the human population because it doesn't affect the germline cells. Okay. But in bacteria, transduction is very important, where a bacteriophage, meaning a virus, can move DNA from one cell into another cell. All right, be able to describe the structure of DNA. What is a nucleotide? There are 4 nucleotides in DNA. You should be able to name them or their initials. There's 4 in RNA. Remember that thymine is not in RNA, it's in DNA, and it's replaced by uracil. Describe how the nucleotides are connected in DNA. Strand, meaning the DNA strand is held together by covalent bonds between the sugar and the phosphate links. So one strand of DNA is held together by covalent bonds. And then the 2 strands of DNA are held together by hydrogen bonds between the nitrogen containing bases. And then the 2 strands of DNA are complementary to each other, where A and the T always bind together, and G and the C always bind together. Okay, and it can be vice versa, meaning C combined with G, as well as G binding with C. Be able to describe DNA replication, discuss the function of DNA helicase, discuss the function of DNA polymerase, what it does, what's the function of the, well, DNA ligase. I may ask you a question about helicase, polymerase, or ligase. Compare and contrast DNA replication between the leading strand and the lagging strand. And then be able to explain why DNA is said to be semi-conservative, DNA replication. Be able to compare and contrast DNA and RNA. There are some differences. DNA has deoxyribose as the sugar. RNA has ribose. DNA has ATGC as the basis, and RNA has T replaced with uracil, or U, T replaced with U. And then they have the same, let's see, G, C, what am I missing? T, C, and T.
A. Or A?
Hmm?
Is it A? They both-
No. Oh, no. Yeah, you're right. It is A. T is replaced with uracil, right? Now I'm getting confused. Be able to compare and contrast DNA and RNA, be able to describe RNA transcription, and then protein translation, discuss what the function of the RNA polymerase is, where it binds to DNA, how it moves along the DNA, transcribing the DNA to make RNA. In prokaryotic cells, you should discuss where transcription occurs. In eukaryotic cells, also where transcription occurs. Transcription only happens in the genes. The material of DNA, which is not a gene, will not be transcribed. You should also discuss what the roles of messenger RNA is, tRNA and rRNA is, and how they react with rRNA making the ribosomes with ribosomal proteins, and how together they all work to perform protein translation. We should discuss what a codon is and what an anticodon is. You should mention what the genetic code is and how the genetic code is redundant, meaning that more than one codon codes for the same amino acid for some, if not most, of the amino acids. And that's what we mean by degeneracy of the code. That there's more than one codon for most of the amino acids. I should be able to compare and contrast bacterial genetics with eukaryotic genetics. Many eukaryote genes have regions that code for the protein or the RNA, which we call exons, and then regions of the gene which do not code for, I don't know, the protein or the RNA, and we call that an intron. Prokaryotic genetics do not have introns. Understand what the lac operon is and what an operon is. An operon is sort of like a super gene encoding the gene of interest, like the lactase enzyme, as well as the operating regions around that structural gene, meaning the lactase enzyme is the structural gene. So to be able to describe the lac operon and how it functions. Understand what a mutation is, know what is a base substitution, what is a frame shift mutation, what's a silent mutation, a missense mutation, and a nonsense mutation. And you should realize that all three of these are a base substitution. Okay. You should also discuss what is a large mutation, a macro mutation. That's not on here. I should put that on here. You should discuss what is a mutagen, a chemical or physical agent that increases the mutation rate. And the DNA always mutates by spontaneous mutation. The spontaneous mutation is just whenever the DNA is replicating and the polymerase makes a mistake. Be able to discuss the following methods for increasing genetic variation and recombination, what's called for crossing over of the chromosomes, what is transformation in bacteria, conjugation, and transduction. And then lastly on this topic, discuss what a plasmid is and what a transposon is.
I have a question, Dr. Gopal. So is there not any introns and exons on a prokaryotic cell genetics?
I should discuss for bacterial genetics because I think the archaea bacterial genetics is more similar to a eukaryotic genetics, but I'm not an expert on archaebacterial genetics because I've never studied it. Okay, but for bacteria, they do not have introns. They only have exons in their gene. Was that your question?
Yes, thank you.
Okay. And for the life of me, I don't remember if archaebacteria have an intron or not. I just don't remember. And like I said, I've never studied archaebacterial genetics. When I went through microbiology, there were no archaebacteria. Tells you how long ago I went through archaebacteria. I mean, how long I went through microbiology. I was in college when they first discovered the archaebacteria. And I took microbiology early in college. All right, let's see. I think that's it for chapter 8. Let's move on to chapter 6, I believe. So in chapter 6 in the lab, you had case studies. So you can definitely count on a case study, as well as a case study is very good for your testing you on your unknown project. I'm going to be testing you on your unknown project. You can kind of get a guess of that. And there's no real way of studying for the unknown project. But if you know how to do a case study, meaning compare different information about the different species and then identifying the species, that will be very good for your unknown project. All right, chapter 5, microbial growth. Understand the physical and the chemical requirements for microbial growth and the associated terminology with that. All microbes have physical requirements for growing, as well as all cells. And that would be things like they have to grow in the proper temperature, the proper osmotic pressure, meaning the proper salinity. There's all their physical requirements. I can't think of any off the top of my head, but there are other physical requirements.
Would pH be a physical requirement?
Say again?
Proper pH.
Yeah, proper pH. Thank you. Like I said, I couldn't think of another one, but there's other physical requirements that cells have to be in order to grow. Okay. And there's also chemical requirements for microbial growth. All life forms have to have access to carbon. Okay. So that's a chemical requirement. I suppose some life forms have to have an energy source that is not chemical, like light, for example. That would be a physical source. But like for us, we have to have a chemical source for getting our carbon and our nutrients, and that would be the food we eat. And then the nutrients are other chemical requirements, and those would be things like phosphorus, sulfur, sodium.
Nitrogen.
Trying to think of some other chemical requirements.
Nitrogen.
Oxygen. Say again?
Nitrogen.
Yeah, nitrogen. That's a very good one. And we have to get that from either the food we eat or the liquids we drink. And that's true for microbes as well. And there are other chemical requirements that they have. There's organic growth factors, and that would be a biological molecule. For us, vitamin C would be an organic growth factor. Okay, any question about that? For some microbes, it might be something like NADH or some coenzyme. Other chemical requirements would be elements, like we have to have a low amount of copper and iron and magnesium in our diet. And most of these elements we get just from eating the food we eat and the water or liquids we drink, and we just need trace amounts of them. Okay, any question about that? All right, be able to discuss the differences between obligate aerobes, facultative anaerobes, obligate anaerobes, aerotolerant anaerobes, and microaerophiles. And you would be able to learn this by looking at the microbe growing in a thioglycolate tube. Where the growth would be in the tube would tell you what the oxygen requirement of the microbe was. Understand the toxic forms of oxygen and why they are lethal to anaerobes. They are lethal to aerobes, it's just the aerobes have the enzymes they need and the molecules they need to inactivate the lethal forms or the toxic forms of oxygen. Okay, like hydrogen peroxide that's made in your cells from simply aerobic respiration. The hydrogen peroxide that's made in your cells is removed by the enzyme catalase. Okay. Understand the importance of the term culture media, including what's a chemically defined media, what's a complex media, what's a reducing media, what's a selective media, what's a differential media, and what's an enrichment media. You should know that an enrichment media is selective by nature, but it's used specifically to enrich for a bacterial species. You should also know that the media does not have to be only one. Like McConkey's media is both selective and differential. And I guess I should mention there's one other type of media that's not on here, and it's a general purpose media. And its purpose is just to grow bacteria cheaply. No selection. All right, understand the importance and be able to describe the process of streaking for colony isolation, getting isolated colonies, and then using that by doing it twice to get pure cultures. Understand the appropriate terminology. All right, be able to describe the process of binary fission and budding in bacteria. Most bacteria reproduce by binary fission, where the cell simply divides in two. A few bacteria grow by budding, and a few, fewer still, grow by forming reproductive spores. I don't know why that's not on here, but you should know reproductive spores. You should know that endospores is not reproduction, because one parent cell makes one endospore and then the parent cell dies. Not reproduction. Understand what bacterial generation time means and how to calculate it. We're not talking about generations. Are we doing that? No, I'm getting confused. We're not talking about generations per hour. It's the hours per generation. That's the generation time.
Is it 20 minutes?
Say again?
Is that 20 minutes?
No, the fastest you'll find in the microbes growing on the lab is that the fastest ones may have a generation time as short as 20 minutes. Most bacteria take longer than 20 minutes to grow, meaning double.
Thank you.
Be able to describe the phases of bacterial growth. Initially, we have the lag phase, then we have the exponential phase, then we have the plateau phase or the stationary phase, and then we have the death of decline phase. Be able to describe the following methods used to measure microbial growth. We can measure the number of microbes by a plate count, also called a viable plate count, because this is the only method that allows you to distinguish between viable cells and dead cells. It uses a serial dilution, and then you either pour that dilution onto a plate or you spread it on the plate, and then you count the colonies that grow up. That's the plate count method. The filtration method is similar, but you pass usually liquid or gas through a filter, and then you put the filter on the plate and allow the colonies to grow. Filtration is only used when the bacteria numbers are very low in number. Like if you wanted to count the number of bacteria growing in the city of Vancouver's water supply, you would probably have to put the water through a filter and then get the colonies to grow that way. And then there's direct microscopic count methods and indirect microscopic count methods. The indirect methods estimate the number of bacterial numbers. The direct method counts the direct bacteria numbers. Like a direct microscopic count, we'll be counting the cells into the microscope under a gridded slide that has a specific volume. An indirect method would be putting a solution of bacteria in a not a spectrophotometer. What do you call that? Spectrophotometer, that's right. And then you measure the amount of light being absorbed, and then you make an estimate of the number of cells. Any question about any of that? Any question about chapter 5? All right. Let's move on to chapter 4.
We haven't done chapter 6.
Did I skip chapter 6? Sorry.
Yes.
I'm not seeing chapter 6. Oh, there it is. Didn't we just talk? Oh, that's why you didn't do chapter 6. It's got chapter 5. I need to correct this. Let me see if I can correct this from here. Obviously, nobody was looking at the chapter 6 study guide and lab notes. Otherwise, you would have reported that there's no chapter 6 study guide and lab notes.
It says chapter 5 objectives, but it has the chapter 6
Notes in it?
under it, or at least that's what it says, but they both say microbial growth.
Let me take a look here and see if I can find. I might have a mistake in the file. There we go. worst comes to worst, I can use that one.
Yeah, I think it's just a typo. I think it's supposed to say chapter 6, because in the book, chapter 6 is microbial growth.
Okay, so you're saying that this should be chapter 6 right there? And
Yeah, I
that
think it
be a
should
6?
say 6. Yeah.
And this is correct.
Yeah,
Okay, I just
because
did
chapter
6 there.
5, chapter 5 is microbial metabolism.
Okay, so, all right, that just needs to be
Microbial.
corrected. Let me make a note of that. Thank you for catching that. All right. Let's move. So I've already discussed chapter 6, right?
Yes.
Okay,
Yes.
so let's close that and go to chapter 5. Chapter 5, microbial metabolism. Be able to define the terms metabolism, catabolism, and anabolism. Understand the difference between anabolic and catabolic metabolic reactions and how they're coupled to each other by the pathway going both ways or by ATP. Understand the concept and components of the metabolic pathway. Understand the role of ATP in the cell. Generally, whenever the cell wants energy to do something, it gets that energy from ATP. So ATP we call the energy currency in the cell. Understand the following concepts and characteristics as they relate to enzymes and the chemical reactions that catalyze. Activation energy. That's the energy you need in order to perform a chemical reaction. The reaction rate, how quickly the chemical reaction occurs. The collision theory is the theory of why the reaction occurs. Two atoms or molecules collide with each other, and if they have enough energy in them when they collide, the reactants will be changed into the products in the collision theory. Enzymes are very specific, and for this class, we're going to say that each enzyme works on one specific molecule. Now, as you know, this isn't entirely true. Like we have certain drugs that mimic the reactant in binding to the enzyme. But for this class to be simple, we're saying that enzymes are specific for one and only one molecule. And that's almost true, okay? It's not quite true, but it's almost true. The efficiency of an enzyme will be most efficient when it's under its optimal conditions, such as its optimal pH, its optimal temperature, its optimal salinity. You should know the naming of enzymes. Most enzymes end with ACE, A-S-E, but not all. And then you should know what an enzyme inhibition is. Okay, you should know that ribozymes are enzymes, but these enzymes are made up of RNA instead of protein. And the only ribozyme we've discussed in the class is the ribosome. When it links, removes the amino acid from the tRNA and links that amino acid to the growing chain of amino acids. That's the only ribozyme we discussed in the class. All right, you should know the categories of metabolic diversity. Cells must get their energy and their carbon source from a specific source, and all cells need an energy source and a carbon source, and how the organisms are classified. They'll be classified as a phototroph if they get their energy from light, and a chemotroph if they get their energy from a chemical. The carbon will be an autotroph if they get the carbon from carbon dioxide in the air or the water, and there'll be a heterotroph if they get their carbon from an organic chemical molecule, such as glucose. Any question about any of that? And then there's more complicated classification. There's the photoautotrophs, the photoheterotrophs, the chemoautotrophs, and the chemoheterotrophs. Okay, and so if I give you a species, you have to tell me, and I give you its characteristics, you have to tell me what troph it is, or I give you the troph. Well, I wouldn't do that. I wouldn't give you the troph and then ask you what the species is. So I would tell you its growth conditions, what it requires for growth, and then you tell me what troph it is. Understand about oxidation reduction reactions. They're always linked and paired. One molecule will become oxidized, the other will become reduced. Understand what they are. The oxidized molecule loses an electron. The reduced molecule gains an electron. And what's an easy way to remember that?
Leo, L-E-O-G-E.
Yeah, Leo the lion says GER, gain electron reduction. Okay. Or Leo the lion goes GER or says GER, one of the two. Leo, lose electron oxidation. So Leo, lose electron oxidation. All right, be able to describe the three mechanisms by which ATP is generated in cells. Substrate level phosphorylation by respiration, and then photophosphorylation. Photophosphorylation only occurs in photosynthetic organisms, but other organisms will get it from either respiration or from substrate level phosphorylation. Here's a trick question. I wouldn't ask you this in an exam, but humans do which of those three? Do they do substrate level phosphorylation to make ATP? Do they do respiration to make ATP? Or do they do photophosphorylation to make ATP?
The first two?
The first two is correct. Good job. Yeah, in glycolysis, we do substrate level phosphorylation to make ATP. And then in aerobic respiration, obviously we use respiration. Understand what aerobic respiration is. It includes 3 steps: glycolysis, the preparatory step in the Krebs cycle, and the electron transport chain, or ETC. You should know what molecules start each step and what molecule ends each step. So when we're talking about glucose, glucose starts glycolysis, it ends with pyruvate. Pyruvate starts the preparatory step, and the Krebs cycle ends with CO2, actually. I don't think we ever stated that. We said the preparatory step ends with lactic acid, not lactic acid. Gosh, what's the name of that? Something coenzyme.
Acetyl.
Isn't it coenzyme A?
Coenzyme A, thank you. And, but it ends with CO2. The Krebs cycle changes the carbon in glucose to the carbon in CO2. And then the electron transport has the NADH and the FADH2 transporting the electrons to the electron transport chain. And then the electron transport chain pumps hydrogen ions across the membrane, which for eukaryotes will be the inner mitochondrial membrane. For prokaryotes will be the cell membrane. And then the cell uses those electrons to make ATP, not the electrons. It uses the hydrogen ions that it moves across the membrane. When the hydrogen ions move back across the membrane, it uses that to make ATP. You should know the number of glucose, the number of pyruvate, the number of ATP, the number of CO2 that are made in each of these steps. Okay. And you should know the ATP molecules involved. It's not necessary to know every single specific detail of respiration, but you should know the summary. So you should know how many ATP are made by aerobic respiration. And for each step of aerobic respiration, meaning there's a net gain of 2 from glycolysis, a net gain of 2 from the preparatory step in the Krebs cycle. And what is it, 34? We do the math here, 2, 4. So 32 to 34 for eukaryotic cells into the electron transport chain, and 34 for prokaryotic cells in the electron transport chain. The total number of ATP made by aerobic respiration is 36 to 38 in eukaryotic cells and 38 ATP per molecule of glucose in prokaryotic cells. Any question about any of that?
Would you ask us about the pyruvate and not the ATP? Like how
I
many?
might ask you how many pyruvate are made by glycolysis starting from 1 glucose.
2.
Yeah, I might ask that. Is that what you're asking? Anyways, I think I answered it, okay? I think I discussed that already. Oh, you should understand the process of fermentation. It starts with glycolysis and then tacks on 1 or 2 fermentation reactions, changing the pyruvate to some other molecule in fermentation. And we've only talked about 2 other molecules. For lactic acid fermentation, it forms lactic acid or lactate. And then in alcoholic fermentation, it forms ethanol. Your cells, your muscle cells can perform lactic acid fermentation as well as aerobic respiration. All right, you should understand the overview of lipid and protein metabolism, because they can be, these molecules can be digested to make ATP, but for most of us couch potatoes, we will convert these molecules to carbohydrates and then utilize the carbohydrate to make ATP. But a marathon runner can digest lipids or fats to make ATP, but they would not do this until after 45 minutes into the run. So cells get their energy, at least cells which are chemoheterotrophs, from a metabolic pathway, breaking down polysaccharides, lipids, proteins, and nucleic acids. Understand the concept of the integration of metabolism. We've discussed metabolism as being separate, anabolism and catabolism, but in reality, the two are linked together by amphibolic pathways. Humans are not very good at linking things together by amphibolic pathway. But as you know, if you eat a high protein diet or a high lipid diet, we will make carbohydrates. So it is connected by amphibolic pathways. And then you eat a high protein diet or a high carbohydrate diet, you know that your body will convert that to lipids and then store it down as a lipid in your belly by an amphibolic pathway. All right, you should describe the two types of photosynthesis. There is cyclic photosynthesis, making ATP, and non-cyclic photosynthesis, making glucose. You only need to know one form of non-cyclic photosynthesis, which is the form which makes oxygen. Okay, you don't need to know the other one. All right, any questions about chapter 5? Not, let's move on to chapter 4. You should know how prokaryotic cells are similar and how they differ. You should know the different terms for describing a bacteria like Coccus, Bacillus, Barilla. Describe the structure and the significance of the prokaryotic glycocalyx. Understand there's 3 types of glycocalyxes, although I'm unlikely to ask you about extracellular polysaccharide. You find that only in, what is that called? Sorry, I'm drawing a blank here. When bacteria cells make a, they put down many layers of stuff around the bacteria, and the bacteria get together all in this stuff, and that is called a, nobody remembers. I'm
Capsid.
sorry, I'm drawing a blank here.
Is
What?
it capsid or envelope?
No. Let me go look down here. I'll find it. It's not a slime layer. It's not a capsule. It's a, why am I not getting this third one? Oh, because this is only the objective. That's why I'm not getting it. Let me go here. Look in the notes.
Biofilm.
Biofilm, there it is. When they're in a biofilm. We didn't really talk much about biofilms. Infections that have a biofilm are much harder to treat than infections that do not have a biofilm. And unfortunately, we didn't go into that in this class. All right, where was I?
Number
Oh,
4.
here we go, capsule. So the other glycocalyx are a capsule. If you can see it under the light microscope, the cell is making a capsule. So the glycocalyx is a capsule. If you cannot see it under the light microscope, we then call it a slime layer. The slime layer is mostly water, and you need an electron microscope to be able to see it. The capsule has more molecules in it other than water. But a glycocalyx can be a capsule or a slime layer and an extracellular polysaccharide, but this last one is only seen in a biofilm. Be able to describe the phenomenon of taxis of bacteria and why it's important. If the bacteria can move by taxis, so taxis is the movement of bacteria in response to a stimuli, it's important to the bacteria because the bacteria want to move to a more favorable environment where it will be able to grow and reproduce more easily. And so that's why the bacteria, taxis is important to the bacteria, to bring the bacteria to a more favorable environment. If the bacteria does not have taxis, cannot move, it's stuck in one environment. And if it can survive, it's just going to have to wait until the environment changes. And that's what endospores do. Okay. They're very good at surviving in a poor environment, and they just wait. Other bacteria can't survive and wait so long, and they will eventually die when in a poor environment. E. coli is pretty easy to kill. Like if the E. coli falls in molasses or into a pickle jar, the E. coli is going to die shortly. I don't know how soon, but it'll be fairly soon. Like in a day or less. I don't know how soon. It might be only minutes. I don't know. Describe the function of flagella. I'm not going to ask you about the axial filament. I probably won't ask you about fimbriae. That's hair-like structures coming out of the cell. A pili, you should know what that is because we discussed that in chapter 8. Describe the structure and the significance of the prokaryotic cell wall and how prokaryotic cell walls can stain gram-positive and gram-negative. And the staining is because of the difference in the cell wall. The gram-positive cell wall is very thick and has a very thick peptidoglycan layer. The gram-negative cell wall has a thinner peptidoglycan layer in the cell wall, and it also has lipids on the outside of the cell wall that we call the outer membrane of the cell wall. You should know what peptidoglycan is. You should know the gram-negative cell has an endotoxin. And this will be the third time we've discussed endotoxins. So you can count on having a quiz question or a final question on endotoxins because we have talked about them 3 different times. I don't remember when we did the very first time we talked about the gram-negative cell wall. And then there was the time we talked about the cell wall in great detail. So we talked about it at those 2 chapters. And I don't remember the third time. Anyways, the difference allows the crystal violet to get stuck in the thick peptidoglycan of the gram-positive cells, and it doesn't wash out. It does wash out of the gram-negative cell walls. And then we stain with the counterstain safranin, which stains the gram-negative cells, which are clear at this point. They will pick up the safranin and stain pinkish. You should describe the structure of the plasma membrane and how it is a lipid bilayer, and it controls what enters and leaves the cell everything that goes through the plasma membrane must be able to go through the lipid of the plasma membrane, meaning the inner part of the bilayer. And anything that wants to move across the membrane, either into the cell or out of the cell, if it can't go through the lipids, then it has to have a transport protein to transport it across the membrane. And this relates to the fluid mosaic model of the plasma membrane. Fluid mosaic model mentions that the membrane is a liquid, and it's holding the cell wall to cell membrane together. And the proteins can move about in the cell membrane unless they're anchored either externally or internally to the cytoskeleton of the cell. And they'd be anchored by proteins. You should know what the term selective permeability means, that some things can cross a membrane, so it's selectively permeable to those items, and does not allow other things to cross the membrane. Be able to describe the following: what simple diffusion, facilitated diffusion, osmosis, what is active transport, what is osmotic pressure, and what is group translocation. I'm not going to discuss all of these now because it'll take too much time, but let me mention that group translocation is similar to active transport, except group translocation only occurs in prokaryotes, not in eukaryotic cells. And when the molecule is moved through an active transport protein to move into the cell, the molecule is physically changed. Like when we move glucose into a bacteria by group translocation, we add a phosphate to the glucose, so it's no longer glucose, it's changed to glucose 6-phosphate. Any question about any of that? All right, you should know what tonicity is and describe what isotonic, hypotonic, and hypertonic solutions are. You should know what cytoplasm is, the bacterial nucleoid region, what's a bacterial plasmid, bacterial chromosome. You should be able to compare and contrast bacterial ribosomes with eukaryote ribosomes. A eukaryote ribosome is larger at 80S. A bacterial ribosome is 70S or smaller. You should know an inclusion or inclusion body is something the cell is incorporated into the cell, and it becomes at high enough concentration that the material precipitates out of solution and forms the inclusion body. You should describe endospore formation and the importance of an endospore for bacterial survival. You don't need to know about the organelles found in a eukaryote. I don't think I asked any question about that on the final, but you might want to know about the organelles found in a prokaryotic cell. And you should know about the endosymbiotic theory. All right, any questions about chapter 4? Let's move on to chapter 3. You should be familiar with the metric system. You should know the size ranges for prokaryotic cells compared to prokaryotic for eukaryotic cells compared to prokaryotic cells compared to viruses. You should know the different parts of the microscope, be able to identify them and say what they do. You should be able to describe the resolution and refraction of a microscope. You should know what an oil immersion lens is and why you need to use oil. You should know what an electron microscope is and that it uses electrons instead of light. You should know why we stain specimens when viewing under a light microscope. You should know how we make a bacterial smear. And you should know that when we make a bacterial smear, we're not using any stains or dyes. Okay. You should know about a simple stain, which when we're doing that, then we take the bacterial smear and we apply the stain. You should know what a mordant is. You should know what a differential stain is. And you should know the Gram stain. We discussed the Gram stain in detail. And the Gram stain is a differential stain. You should know the difference between acidic and basic dyes. In a simple stain, there's a simple basic stain, and then there's a simple acidic stain, which we can also call a negative stain. And the negative stain or the acidic stain stains the background of the cell. It does not stain the cell. I already mentioned the Gram stain. And then you should know what the color of a cell is at each step in the Gram stain. Any question about study chapter 3? If not, let's quickly go over chapter 2. I'm unlikely to ask you specific questions about chapter 2. However, I will take it that you understand the chemistry of chapter 2, meaning when I ask you a question, it's understood that you understand the chemistry. So you should know about chemical bonds, covalent bond, an ionic bond, and hydrogen bond. You should know about water and the properties of water and how they're important for life. You should know about pH. I don't think I'm going to ask you about that. All right, you should know the 4 biological molecules. The molecules of life include the carbohydrates, the lipids, the proteins, and the nucleic acid. If I give you one of these molecules, you should be able to tell me what molecule it is, a carbohydrate, a lipid, or a protein, or a nucleic acid. All right, any questions about chapter 2? All right, that was pretty quick. Chapter 1 goes over the history of microbiology. Do I like the history of microbiology? Anyone? Okay, nobody's answering. I like the history of microbiology, so I'm more likely to ask you about a question on chapter 1 because it's talking about the history of microbiology in very quick and short details. Okay, so you should probably study the major historical events in microbiology, who did them and what they are. Like Louis Pasteur invented pasteurization. He also invented aseptic transfer techniques. You don't need to know when he did that, but you should know that he did that and they were important in the history of microbiology. Edward Koch is another one you should know, and he did a number of things as well. I only mentioned some for Louis Pasteur. He's got a whole bunch of things that he did. He did pasteurization. He did fermentation. I don't remember all the things he did, okay? Dr. Koch, the main events he did was he did Koch's postulates, where he came up with a procedure to identify a microbe as causing a specific disease, as well as he came up with the first microbe that was shown to cause a disease. And that was Bacillus anthracis, causing the disease anthrax. He also came up with solid media, and then made a very easy way to get a pure culture by streaking the cells over a plate and getting isolated colonies, and then doing it 2 times before you set up your stock culture. All right, any question about any of that? So I like the history of microbiology. You should study it. I'll probably have more questions on that than other chapters. So most of the other chapters they haven't been quizzed on, I will only ask 1 or at most 3 questions on. For chapter 1, I will ask more questions on. So you should know what a microbe is and a microorganism, know what a pathogenic microorganism is. You should discuss the beneficial roles of microbes on both the earth and to people, like they're part of our microbiome, what the normal microbiota is, where it's normally found and where it's not found. So it's found on all external tissues and all of the mucous membranes open to the outside. But the normal microbiota is not normally found internally, such as the heart, the liver, the blood inside the body, and bone, stuff like that. I've already talked about historical things. You should know about the first vaccine in Western medicine, the development of the vaccine to prevent smallpox. And generally that was vaccinia, which is where we get the term vaccine, but it protects against smallpox from using cowpox. Discovery of fermentation, we talked about that at least twice. There's another thing that Louis Pasteur did. It's important that he's the one who disproved the theory of spontaneous generation, that microbes could spontaneously generate when a vital force came into something, such as you throw the log into the fire and the newt crawls out. It wasn't from spontaneous generation of the log, the wood being thrown into the fire. But the newt came from pre-existing life, and that's the theory of biogenesis. All right, here's some of the things that were important in the history of microbiology: the development of the aseptic technique by Louis Pasteur, pasteurization by Louis Pasteur, the idea of aseptic surgery, which Louis Pasteur did not come up with that because he was not a doctor. He did come up with the idea of aseptic technique, and then that was used for aseptic surgery. And the person who really proposed it and got it to stick was, oh gosh, it's not Jenner. Who the heck is that? I'm gonna have to look that up. I don't remember. Anyone remember? Actually, I might be able to do this. The phenol has a coefficient of 1, and we call that coefficient. Nope, not able to do that. I'll look it up. Cox postulates I've already talked about.
Joseph Lister?
Say again?
Joseph Lister?
Lister, thank you. Lister's coefficient. Yes, Joseph Lister was the one who developed aseptic surgery and got it to stick. There was somebody before him who tried to get it going, but he was actually fired. I don't remember, it was the vice president or the president of the hospital was insulted that this other guy was proposing that the doctors wash their hands before performing surgery. And so he fired the guy. But Joseph Lister was the one who got it to stick. And he showed that when you perform surgery under aseptic conditions, the patient is more likely to survive the surgery or from the infection, because there's not going to be the infection after the surgery. I describe what an infectious disease is. You probably don't need to know about an emerging infectious disease. You should know what a virus is, because I would study that later in the prion. And I think that's about it for chapter 1. Any questions?
There was a little bit on this section about chapter 10. What should we know
Yes.
on chapter 10?
Chapter 10, we discussed mostly the classification. Where do I have that?
All the way down at the bottom, I think.
It's probably on another one.
It's on
I
the
don't
same.
see it here.
Oh, it's on the same.
Oh, it's only there on chapter 1. I don't say chapter 10. Oh, there it is there. Yeah, it is at the bottom. The vocabulary of the chapter, know why organisms are classified into 3 domains: the tree of life, the archaeobacteria, the bacteria, and the eukaryotes. And we define the species by sequencing the DNA, and depending on the sequence of the species by their DNA, we can determine which domain it goes to, so that the domain is actually defined for which it is. The kingdom is a organization underneath the domain. The kingdom is not defined, and people argue about which kingdom an organism goes into. And because it's not defined, it's, well, I guess whatever you want, okay? Like some people consider algae, especially kelp, to be plants, and other people consider it to be protists or some other kingdom, okay? And because the kingdom is not defined, it's open for interpretation. You should know about the binomial nomenclature and how we name organisms using 2 names. The first being the genus name, and then the second being the specific epithet. And you need both to name the species. For bacteria and eukaryotes, all of these names are Latinized, but for viruses, we actually use common English names. You should know how prokaryotes are classified and how eukaryotes are classified. The main difference being the nucleus found in a eukaryote. But eukaryotes also have membrane-bound organelles, which are not found in prokaryotes. All right, any question about chapter 10? That was a good question, by the way. I sort of overlooked chapter 10. All right, if not, we're done. If you have any questions, email me.
Thank you.
and
Thank you.
Hope you do well on the final. All right,
Thank you.
Thanks.
bye.
Bye.